Be careful! No COVID-19 vaccine is 100% effective.
Natural and vaccine-induced immunity might not last forever.
The next step is to measure the effectiveness of COVID-19 vaccines. Every immune system is different. It depends on different factors, such as age, genetics, underlying medical conditions, people with cancer, chronic diseases, etc.
That is why detection and quantification of the neutralizing antibodies against SARS-CoV-2 are important.
What are the neutralizing antibodies? The neutralizing antibodies protect us from getting infect from SARS-CoV-2. Entry of SARS-CoV-2 into human host cells occurs through binding of surface unit S1 of its spike protein to the cell receptor ACE2, which leads to viral replication and then spreads SARS-CoV-2 infection. Such infection typically induces antibody response. The antibody is able to bind to the SARS-CoV-2 spike, and only a small part of the antibody with neutralizing function. Only neutralizing antibody is capable of blocking the action of SARS-CoV-2 spike and inhibits the SARS-CoV-2 virus to infect new cells. The neutralizing antibodies are generated naturally by the immune system after getting infected. COVID-19 vaccines help our bodies develop immunity to the virus that causes COVID-19 without us having to get the illness. There are 3 methods to quantify and detect the neutralizing antibodies. First method, virus neutralization test, VNT, which is risky because it requires live cells and live SARS-CoV-2 virus. Besides that, a specialized biosafety level 3 BSL-3 containment facility is required. Second method, PSEUDOVIRUS-Based Virus Neutralization Test, PVNT. It requires complex equipment and reagents. This method is quite slow. The whole process can take from 2 to 4 days. Third method, MaxNAb SARS-CoV-2 Neutralizing Antibody ELISA Kit. It is safer and easy to use ELISA kit. No dangerous cells, nor live SARS-CoV-2 virus required. Extremely fast, requires only 50 minutes to process. Strip-well microplate format allows you to choose the manual or high-throughput analysis. MaxNAb is highly sensitive and specific. MaxNAb can detect neutralizing antibodies as low as 1 ng. It is specific for SARS-CoV-2. It distinguishes NAb from other antibodies. High quality, made in the US.